All right, so in this section, we are going to be taking a look at some of the chemical properties or the chemical characteristics, which will eventually lead to the chemical reactions of our triglycerides. And the chemical reactions that these undergo are going to depend upon the types of functional groups present. And we've seen these two functional groups before. We had our carbon-carbon double bond that we've seen with our unsaturated fatty acids. And then we're also going to see our ester bonds, which have that C double bond O. So we have looked at reactions of alkenes, which have carbon-carbon double bonds previously in chapter 11. And in the previous chapter, we looked at the reactions that the esters will undergo. Now we're going to see those same reactions in our triglycerides. So let's first of all take a look at what can happen to those double bonds. And the first thing that we're going to take a look at is adding hydrogens to those double bonds, right? And typically we said that double bonds are found in your unsaturated fatty acids. And in a hydrogenation reaction, right, that here tells us that we're adding hydrogens. And specifically, we're going to add hydrogens to that double bond. And we've seen that before in the alkenes chapter. So as a result, when we add hydrogens to that double bond, the double bond gets converted to a single bond. So in other words, we go from a unsaturated to a saturated compound. Right, and as we saw previously, when we looked at hydrogenation reactions of alkenes, those typically happened by adding hydrogen in the presence of some sort of a catalyst, which we've seen as either being nickel, or sometimes you'll see palladium, PD, or platinum, PT. Right, and that's your catalyst. That's the thing that speeds up the reaction. So we have our double bond over here. We break that double bond, we convert it to a single bond, and then we just add a couple extra H's to make sure carbon has 4 bonds. So if we have a triglyceride like this one here, glyceryl trioleate, and we hydrogenate it, right? So again, you want to look at your reactants and any special conditions. So the fact that we're adding hydrogen in the presence of a nickel catalyst, that is your indication that it is hydrogenation, right? And this will happen at the double bonds, right? So now we can know, or we should know, where to focus in our molecule. And specifically, it's the carbon-carbon double bond that we're looking at. Right, so if we look at our structure over here, right, we can highlight where all the action is going to happen, right, which is going to be just at those double bonds. So we end up converting those double into single bonds, right? So we end up with a completely saturated triglyceride. So the one that we started off with would have been unsaturated, and the one that we end with here is going to end up being saturated, right? So it's just an application of the chemistry that we saw back in chapter 11, where we are going to be adding hydrogens to that double bond. We convert it to a single bond, so we go from an unsaturated compound to a saturated one. So this reaction is performed on a huge scale, commercially, in other words, and what it's done, or what is accomplished here, is on a commercial scale, liquid vegetable oil, for instance, which has lots of double bonds in them, they become hydrogenated, but not hydrogenated all the way. They actually stop the hydrogenation before all of the double bonds are converted to single bonds, so we end up with partially hydrogenated vegetable oils. And as a result, the consistency of the thing changes. So it goes from being a a liquid, which is what an oil is, and we end up making slightly different looking things, like your soft or semi-soft, solid, or fatty looking things, which obviously have a different texture associated with them, right? So it becomes more saturated as a result of adding more hydrogens, and as a result of that, it's going to have a higher melting point. It's less like a liquid and more like a solid. As a result of hydrogenation. So let's backtrack a sec and take a look at when we did have our unsaturated fatty acids, which again contain your carbon-carbon double bonds. There's a possibility of the groups attached to that double bond being on the same side, which would be cis, or being on opposite sides of the double bond, which would be trans. All right, so let's just draw in the H's, for instance, that are attached to these carbons here of the double bond. So there'll be an H attached here and an H attached to that carbon, right? So if you were to draw a line right through a double bond, you'll see that both of those H's are on the same side. So that would be cis. If we had our one underneath here, you'll see that the H is coming up from that carbon and the H is going down from the other one. Whoops. And if you were to draw a line right down the double bond, you'd see that these are on opposite sides, and this would be a trans isomer. So what can happen during this process of hydrogenation of the unsaturated fatty acids? We said that double bonds get converted to single bonds. But what happens also as a side effect of this is that a small number of the cis double bonds, which remember are naturally occurring, so these naturally occur in the cis conformation. These actually get converted to the trans conformation. And that causes a change in the overall structure of the fatty acid, right? So remember in the cis, we saw this in the previous section, right? So cis double bonds leads to a spreading out of the carbon tails, right? So they don't pack close together. And as a result, it's more like a liquid. Well, when you have that trans double bond, it actually resembles in structure a carbon-carbon single bond. Single bond, which means there is going to be close packing of the tails. Right, and as a result of that, they are going to behave like your saturated fatty acids. All right, and what they actually do, these trans fats, or these trans fatty acids, they actually raise a particular type of cholesterol known as the LDL cholesterol, which stands for low density lipoproteins. So LDLs, and you can consider these, you probably heard the term bad cholesterol. And they lower the good cholesterol, which is the HDL, or the high density lipoproteins. right? So they actually raise the bad stuff, these trans fats do, and they lower the good stuff. So overall, they're not necessarily good for you, right? So if we were to summarize what occurs when a cis fatty acid gets hydrogenated, all right, so this is the tail here, the fatty acid tail, right? There's a kink in the tail due to the cis double bond. So there it is, right? And the H's on the same side make it a cis double bond, and it has this little kink here, right, in the tail. We hydrogenate it with hydrogen and nickel, and what happens is we can convert it completely to the saturated product, right, all carbon-carbon single bonds. But what can also happen as a byproduct is the cis double bond gets converted to a trans double bond, a trans fatty acid. And if you look at the structure, right, they're very similar to each other between the trans and the saturated fatty acid. So foods that contain that naturally occur that contain trans fats include your milk, eggs, and beef. But foods that contain trans fatty acids from the result of hydrogenation include things like your vegetable shortening, your soft margarines, crackers, potato chips, etc., cookies, and deep fried foods. Right, so these have trans fats that result from that hydrogenation process. So that's the first type of a reaction that your triglycerides will undergo due to the presence of the carbon-carbon double bond. The second functional group that's present that will dictate the type of reaction that they're going to undergo is the presence of the ester bond. Right, remember, we can also call your triglycerides as tri-esters because they do have 3 of the ester functional links in their molecule. And when we looked at esters in the previous chapter, we saw that esters typically could undergo hydrolysis reactions, right, where we split that ester with the help of water, either under acidic or under basic conditions. And the products that we get ended up being slightly different in each case. So when we hydrolyze a triacylglycerol or a triglyceride, it is going to split into its components. And the components, the alcohol that we have, would be the glycerol, right? That's your alcohol that we use to make the triglyceride in the first place. And then the carboxylic acids were the fatty acids. Right, so we can say then that your triglyceride can undergo hydrolysis to then produce glycerol, the alcohol, and 3 fatty acids. So the only way we're going to get these specific types of products is if we subject the triglyceride to water and an acid catalyst, right? So think about that for a second, right? The acid catalyst that we normally saw before was sulfuric acid, which is a very, very strong acid. And you wouldn't want to, let's say, ingest that because that would be counterproductive to keeping you alive. But what your body does is it actually has some molecules that would do the same thing that your strong acid and water would do, and that would be an enzyme. Right? And these enzymes that break down these lipids are called lipases. So they would substitute for the strong acid and water, because we want to avoid having strong acids in the body, for instance. So these biological molecules, or enzymes called lipases in this case, are able to do the exact same thing. So we find these in living organisms, including yourself. And the products here that we're going to get are going to be the same thing, glycerol and 3 fatty acids, right? And think about it, your triglycerides are found in lots of things that you eat and drink. And in order to reap the benefits, the energy benefits of that, you should be able to digest those things that you eat and drink. So if we look here again at our triglyceride, right, we can identify where our ester bond is in each case. And just like we've seen before, we were able to split those ester bonds right after the carbonyl in each case. And we end up now on the left side of this with the alcohol portion, which is going to be our glycerol that we make. And then we just convert each of these carboxylic acids here, we convert them to the corresponding carboxylic acids, right? We just add that OH at the end there to make that carboxylic acid functional group. This is the exact same thing that you did in the previous chapter when you hydrolyzed an ester under acidic conditions. We're saying now the same thing's going to happen in the body if we use an enzyme called lipase. We also saw that hydrolysis of esters could be completed under different conditions. So for instance, ester hydrolysis under basic conditions, using a strong base, for example, right? We saw that as well in the last chapter. We called that type of a reaction, that type of hydrolysis, we called it saponification, right? Because now we can see where that term actually comes from. The basic hydrolysis of your fats, for instance, the special type of triglycerides that have these saturated fatty acids, that's actually how you make soaps. Right, so saponification, the reaction of a fat, right, which again is just an example of a triglyceride with a strong base like sodium hydroxide, or sometimes you'll see potassium hydroxide being used in the presence of heat. And it's going to hydrolyze or split the triglyceride. Again, we're going to get the alcohol, which is glycerol, and now instead of getting the carboxylic acid, we're going to get the salt of the carboxylic acid, right? In this case, if we use sodium hydroxide, we'll get the sodium salt of that carboxylic acid. And that essentially is the process that's used to make soaps, right? And the soap molecule, what it is just a salt of a fatty acid. And a fatty acid is just a long chain carboxylic acid. So if we use sodium hydroxide for this saponification reaction, we get more solid looking soaps that can be molded into different shapes. If we use potassium hydroxides, then we get more softer and liquidy type soaps, right? So depending on the type of base that you use, the type of soap that you produce is going to be slightly different. So let's take a look here at our general reaction. So our fat or an oil can be reacted with a strong base in the presence of heat. We end up making the alcohol and then the salt of the carboxylic acid. All right, so this fat or oil here is our triglyceride or tri-ester. We have our strong base, so this could be NaOH or KOH. We get our alcohol, which is glycerol, and then we get the salt of the carboxylic acid, which is the fatty acid. Right, and this is the important product, that's the soap. So again, if we look at our triglyceride or tri-ester, locate the ester functional group in each case, right? The C double bond O is going to tell you where that is. We're going to split it right after, immediately after the C double bond O in each case. All right, and then the other side of the molecule here, right, you can see your 3 carbon alcohol, that's your glycerol that we make. And then the remaining section, the other parts of it here, now we're going to get the sodium salt of each of these carboxylic acids that are going to be in purple, and we convert them the same way that we did in the previous chapter, right? So these are our sodium salts of the carboxylic acid. That's your soap that you end up making. Again, the exact same reaction that you saw in the previous chapter. All right, so let's just finish up this subsection here by just recapping really quick, taking a look at an example. So it says to write the equation for the reaction catalyzed by the enzyme lipase. So we're looking here now at the hydrolysis of a triester that hydrolyzes glyceryl trilaurate, and we're given the structure of it here during the digestion process, right? So the key thing is we're told that it's a lipase, and then also it's hydrolysis. So first of all, we're going to locate the ester bonds. Okay, so we'll highlight those. There they are. Okay, we're going to split them. Oops. We're going to after the C double bond O. All right, so we're going to break it right here. We're going to break that one. We're going to break that one. All right, so now we can see what the products are going to be. On the left side here, we have the thing in green. So let's draw all of that. That doesn't change. So we have a CH2, this should be CH. And then we have a CH2 down here at the bottom. Okay, and then all we're going to do to make it into glycerol, we're going to add that H from the water molecule. That's our glycerol that we end up making. I'm going to draw the H's in a different color here. Let's do the H's in red, just to show you how they change. And then the other product now think about this under the using lipase as the enzyme, we should also get the carboxylic acid or the fatty acid, right? So those are the products for lipase digestion. So let's just see what we have, and we're going to draw each of those individually. So we have this fatty acid here in orange, we have another one, and we have another one. So if you look, right, they're all the same. So we have 3 of the same fatty acid. So what I can write then is 3, and then I'll just draw one of those structures. So we have, again, draw everything that doesn't change. So we have a CH3 on one end. We have a CH2. We have 10 of those. Remember, that's what that notation means. And then it ends in a C double bond O, right? And in order to convert it to the carboxylic acid, we just add that OH. All right, so we have 3 of those because those are the 3 fatty acids that we used. All right, and that's the other product, the second type of product that we get in this process.